Hi, I'm Blair Holman, and my project is the controlled apoptosis of cancerous cells via a 3-plasmid system. The end goal of my project is to detect for tumor cells with high levels of BCL2 and induce apoptosis, eliminating cancerous cells. This would be done via an engineered bacteria with a biosensor with a 3-plasmid system that will detect for the high levels of BCL2. My motivation is to cure cancer. Cancer is the number 2 leading cause of death in the US, with lung cancer, prostate cancer, breast cancer, and chronic lymphocytic leukemia having some of the highest rates of death. Each of these types of cancer has BCL2 in common, an anti-apoptotic protein. BCL2 inhibits the Bax pathway in cells and is correlated with the downregulation or deletion of microRNAs 15A and 16-1. These last two properties of BCL2 I will go into in more detail in the next few slides. Here is a brief schematic of the Bax pathway, which is the apoptotic pathway that BCL2 inhibits. Bax allows cytochrome C to exit the mitochondria, which creates a caspase cascade, including caspases 9 and 3. Caspase 3 initiates apoptosis via DNA fragmentation and chromatin condensation. When BCL2 binds with Bax, cytochrome C is inhibited from leaving the mitochondria, thus preventing apoptosis and promoting cell proliferation. My project aims to create an engineered bacterium that would ultimately detect for levels of BCL2 and initiate apoptosis in cells with high levels of BCL2. Here I show the correlation between BCL2 and microRNAs 15A and 16-1. When BCL2 levels are high, there are no microRNAs 15A and 16-1, and vice versa. As I previously mentioned, I'm detecting for high levels of BCL2, which I would do through detecting for microRNAs 15A and 16-1. So I expect to see no microRNAs when BCL2 is present, as in cancer cells. My genetic circuit will be comprised of 3 plasmids. The first plasmid detects for cell density, as has been previously published. As the cell density increases, AHL accumulates and above a certain threshold activates the Lux regulator, which will induce expression of invasin under the control of the genetic circuit. Invasin allows the bacterium to enter the cell, which activates the second plasmid. The second plasmid would contain a padlock probe, PPAD-MIR, that will respond to the invasion and bind to its complementary microRNAs. The third plasmid would contain the killer gene CCDB3, which responds to unbound PAD-MIR and causes cell death. When there are no microRNAs 15A and 16-1, as expected in the cancerous cells, PAD-MIR 15A 16-1 would not be bound and the killer gene would be activated. This would result in apoptosis of the cancerous cells, eliminating cancer. To get into the basics of my methods, I will be using top 10 F0 cells for the cell growth of 100 cells, 50 healthy and 50 cancerous. The created plasmids would be maintained with chloramphenicol and kanamycin. I would use E. coli strains for the engineered bacteria and genetic circuit, which will be kept in liquid media and supplemented with antibiotics. I will not go into my methods of designing and creating the plasmids here, but I will say what they are for. The first plasmid, PAC LUX-INV, would contain the invasin gene and LUX promoter, initiating the invasion of the cell by the bacteria. The second plasmid, PPAD-MIR, would contain both padlock probes for microRNAs 15A and 16-1 and detect for levels of these microRNAs in the cells. The third plasmid would contain the killer gene CCDB3 and the padlock probes, which would be activated upon recognition of no microRNAs. Here I modeled the kinetic events in the circuit function. My differential equations are as follows. Equation 1 models cell growth and death. Equation 2 models the AHL signal. Equation 3 models the production and degradation of invasin. And equation 4 models the production and degradation of the killer protein. The values I used were either obtained from previous studies or from biological information. Here is a graph of my model results. The blue line, representing cell density, is exponential for the tumor growth over time. As cell density increases, the green line AHL is also increasing. When AHL hits its threshold, the red line of invasin is activated and increases in concentration. Once invacin is high enough for the bacteria to enter the cell, and the model predicts for no microRNAs to be detected, the killer gene E will increase to cause apoptosis, as shown by the purple line. The results of invacin are expected to be the same as previously published. Figure A shows the expected results of the experimental cell culture where the cell is invaded at the threshold cell density. Figure B shows results from my model with 50 healthy cells and 50 cancerous cells. The cancerous cells are red and the healthy cells are white. 100% of the cancerous cells are expected to be invaded, as shown by the X. To confirm the correlation with BCL2 and microRNAs 15A and 16.1, experiments would be done to test for levels of each. The results would be as shown, with levels of BCL2 reduced when the microRNAs are present and vice versa. Prior to putting the padlock probes into the engineered bacterium, I would test for the padlock probe detection of the microRNAs. I would expect the results to be as figure C shows, with the padlock probe for microRNA 16.1 detecting for the microRNA correctly. Thus, I would expect the padlock probe in the cell to detect for levels of the microRNA. Figure D shows results from the model, with the boxes representing invaded cancerous cells turning blue when no microRNAs are detected. I expect 100% of the invaded cells to detect no microRNAs. Lastly, I expect 100% of the invaded cancerous cells with no microRNAs detected to undergo apoptosis via the killer gene, as shown here by the black boxes. Apoptosis assays would be performed in experiments on the transfected cells with the plasmid containing the pad MIR15A 16.1. So all of the cancerous cells are expected to die, thus eliminating the cancer and completing my project goal. Thank you for your time.